hemisphere who are at home watching this in their pajamas will now not be able to see. Please note that the instruction card on your chairs or the sign up by the stage, this one right here, gives you the instructions, and you just simply text Mercedes using this quasi-phone number 25827, and then you will receive a reply thanking you for joining, and then you can ask your question on your smartphone. If time does not permit, since this is a live broadcast and it can only last, 30, 45 minutes, you guys, as the lucky folks who have a chair here, will be able to talk face-to-face, live and up close with both Doctors Naoki Nakayama and Dr. Brian Davis, and they are happy to join you afterwards and be able to talk to you in person about whatever questions you have. So I think that about wraps it up. Thank you all, and we hope you enjoy this evening. Okay.
Hello and welcome to UTHealth House Calls, presented by Mercedes-Benz Dealers of Greater Houston. My name is PJ Afrasiabi, and I'm the General Sales Manager here for Mercedes-Benz of Houston Greenway. Thank you for joining us tonight of number 6 of 7 house calls hosted throughout the area of covering a variety of health topics. We are proud to partner with UTHealth as we bring awareness to important health issues and incredible work that's being done by healthcare educators, providers, and researchers at the university. Thanks to the partnership, I'm also proud to announce Mercedes-Benz of Houston Greenway has given $5,000 scholarships to two students of McGovern Medical Schools at UTHealth. Tonight's program, The Future of Stem Cell Therapy, is presented by Dr. Brian Davis. Dr. Davis is the Director of Center of Stem Cell and Regenerative Medicine and Associate Professor of Molecular Medicine at Brown Foundation Institute of Molecular Medicine at McGovern Medical School. He's also a member of the Genetics and Epigenetics program as well as Immunology program at the MD Anderson Cancer Center of UTHealth Graduate School of Biomedical Sciences. So the smartest guy here. The Q&A portion of tonight's presentation will be moderated by Karen Kaplan, a medical journalist and a longtime member of the UTHealth community. Once again, and thank you for joining us at Mercedes-Benz Houston Greenway, and please join me in welcoming Dr. Brian Davis.
All right, thank you for being here tonight and for watching online. What I'd like to do is just within about 15 minutes is give you a little bit of background and perspective on stem cells, particularly as we think about using them therapeutically, both now and since it's titled The Future of Stem Cell Therapy, trying to give you an idea where we're going. Just to set the stage, why do we need stem cells? And it turns out that our bodies, especially our actively regenerating organs like the blood system, the skin, the intestine, they need the cells on an ongoing basis to be able to replace cells that are lost due to death or to some kind of injury. So for example, our skin is essentially replaced about every 28 days. So external skin is essentially replaced, and it's in fact stem cells that sort of sustain and regenerate the new layer of skin that emerges. In the case of the intestine, it's the internal cells that sort of are in the inner lining. They're replaced about every 2 days. So if you think about how often during the course of life one needs to regenerate those cells. If we look at the blood system, there's ongoing production of blood cells throughout life coming from what we call the marrow which is in the bones. Ongoing production of cells. And just to give you an idea of the level of production, if I only take just one type of those blood system cells called an eosinophil, that's involved in what we call the innate immune system that recognizes microbes and infected cells and eliminates them. We're in fact every second generating 1 million new eosinophils. So if you look over the course of a lifetime, that is the equivalent of about 3 times 10 to the 50 to 3 quadrillion. So 3 million, thousand New cells. And if you just think about what mass that is, it's essentially the mass of 2 C-Class Mercedes out in our showroom here. If we think about an S-Class, it's going to be maybe 1 1/4 or so. But it's a lot of cell mass that's needed to be regenerated. And organs like the lung, which don't turn over that rapidly, but we have stem cells within them called basal cells that in the case of injury, that can replenish the surface cells of the lung. So stem cells are essentially cells that not only can turn into more functional, or we call differentiated cells, but they can actually make copies of themselves, so that there's a way, if you will, for the internal factory of these organs to continue to always make sure that they have some of these long-lived stem cells to regenerate. So if you think about using these therapeutically, in the case of the blood system, we've been using essentially stem cells in therapeutic use for about 4 decades now. So for instance, patients who are, let's say, treated with high-dose chemotherapy for certain cancers, usually these reagents, these chemotherapy agents, kill dividing cells, so they tend to kill off the blood system. So what will happen is after the patient is treated with that high-dose chemotherapy, they're given an infusion of new blood stem cells. And those cells are capable of repopulating the full blood system of the individuals who would have lost the cells from the chemotherapy. And these stem cells are have such a potential in terms of giving rise to cells that if you take, for instance, a mouse that's maybe 1/1000th of our size, in that case, 1 blood stem cell is enough to repopulate the whole mouse. So if we do the same high-dose chemotherapy in an animal, you give them, so it has tremendous potential in doing so. So these blood stem cells have already been used for high-dose chemotherapy. They've also been used for children born with certain inherited immune deficiencies or DNA damage syndromes. And essentially, in that case, what you need is a closely matched donor. You need either a sibling or someone who's very, very closely matched so that when you deliver those stem cells and they produce the blood cells, they'll not be rejected. So one type of stem cells that we think about for therapeutic uses are what we call these tissue-resident stem cells. These are the ones like the blood stem cell, the skin stem cell, the lung stem cell. These are ones that we can think about using therapeutically to essentially repair damaged organs or destruction due to disease or injury. So that's one class of stem cell. Another type of stem cell is one that is really not used to itself replace the damaged or injured cells, but it really plays primarily an anti-inflammatory role. And these are cells called mesenchymal stem cells or bone marrow stem cells, not the blood stem cells that are in the marrow, but these is another class. And these cells can, in their normal function, give rise to either fat or muscle, bone, cartilage. So what people began to do maybe 10 years or so ago is they began to look in the case of, let's say, heart attacks. And then more recently, we have colleagues at our institution, Dr. Charles Cox, who's using these for traumatic brain injury, a colleague, Sean Savitz, who's using them for stroke. And essentially, one delivers these cells either through the bloodstream or maybe directly into the brain. And in this case, the cells, they're not replacing the damaged neurons or the damaged structural cells in the brain. Really, they have an anti-inflammatory role. They're basically trying to, normally our bodies, when they experience injury, have a natural response, which is try to flood the system with cells that sort of help begin repair. But then sometimes things go awry and there's too much inflammation going on. And what people have found is that these are the cells that can help reduce that inflammatory process and therefore help the body itself begin the repair process. So not replacing the neurons or not replacing the cells damaged, but rather this anti-inflammatory role. These, in fact, are the cells that are primarily, if you see advertisements either in the newspaper or on the web, or you drive by certain highways here in Houston where you'll see signs for stem cell clinics. Those are often the types of cells that are being used in that regard. That is, cells that are obtained either from a patient's bone marrow or even from their fat tissue, or from, let's say, umbilical cord blood or umbilical cord at the time of delivery. So that's typically the types of, and we can come back to that in the question and answer session. Let me talk about now the type of stem cell that has received a lot of interest in the last 10 years since they were first discovered by a Japanese scientist, Shinya Yamanaka. And basically, what we know is that once once an egg is fertilized with a sperm, and the process has begun of sort of natural development of a new human being, for example, what we know is that in some of those early stages, there is the formation of tissue types and cells, and they're a particular type of cell that can give rise to every cell type in the body. From those cells give rise to the bone and the tissue and basically what people thought is this is really a sort of a cascading one-way direction that as you go from this very unspecialized stem cell to the more differentiated or mature cells. You start from this undifferentiated cell and it becomes either a blood cell or a neuron in the brain or a skin cell. People thought that that was just a one-way process. And essentially the genius of this scientist from Japan, even though he's really a genius in what he does, he's a very humble person. You know, when you hear him speak and answer questions, it's just really a very good individual. And what he asked the question, can we go the other direction? Can we start with a skin cell, or can we start with a blood cell? And can we go upwards? Can we essentially take a cell that's already been committed to a certain function in the skin, or let's say one of these eosinophils that has this particular function in the innate immune system, can we somehow take those cells and convert them into a cell which has all the properties of these very undifferentiated, non-specialized cell, but with this tremendous potential to give rise to all the cells. And the amazing thing is that back in 2006, first in the mouse, and then in 2007 in the human, he showed that he could do that. It turns out that one doesn't change the genetic makeup of the cells as you convert that. You simply change the sort of the structure of the chromosomal DNA, and regions that were previously closed and couldn't make proteins now begin to make proteins, and other areas that were making proteins get shut down. So you take a specialized skin cell and you convert it back into these cells that have all this potential. So I don't know if you recall, but back in the time of maybe 2003, 2004, there was a lot of issues that went on regarding, are we going to be able to use, quote, embryonic stem cells? Because there were a lot of moral and ethical issues regarding how one obtains those cells and how they'll be used. And it turns out that essentially through what Dr. Nakayama discovered, we can in fact create cells today from any individual in this room. I say in any individual in this room because they've shown that they've done it, I think, up to maybe age 95 individuals thus far. You don't have to be a young person to contribute tissues to sort of reprogram. And what was shown is that you can in fact convert these cells to cells that in the tissue culture dish look like embryonic stem cells, although they never involved an embryo. They came simply, they were sort of what we call reprogrammed up to this highly potent, and in fact, just within our center at UTHealth, we've converted those cells into blood cells, cartilage cells, bone, muscle cells, neurons, glial cells, lung cells, all starting from that population of cells. So in principle, what this means is that because, let's say at some point, you were to have a sports injury that you damaged the joints of your knee, or maybe just through aging, just through normal processes of life, that you began to wear away all the cartilage and you begin to get bone on bone in terms of the knee. Well, in principle, and we're not there yet, but for instance, Dr. Nakayama, who's helping out with answering the online questions tonight, he's a pioneer in the world in terms of actually taking these, what we call induced pluripotent stem cells, and turning them into cartilage that, if it can be scaled up, have the potential of delivering cells back to your body, even tissue back to your body, that will not be rejected. Why it will not be rejected is because those stem cells, and thus the cartilage, were all derived from your own genetic makeup. There's no rejection going on. In my laboratory, we're trying to use these same induced pluripotent stem cells. All by induced means we've sort of induced a process. We sort of stimulated a process. Pluripotent is just a word that means many potential, which means it can make all the cell types that I spoke about, essentially the neurons and the lung cells and the skin cells, liver cells, or anything that you want to create, as long as you have the right recipe or protocol in the laboratory. And so what we're doing is we're actually using this primarily for individuals with inherited genetic disease. And what do we mean by that? We mean, for instance, in the case of cystic fibrosis, it's a disease that affects individuals throughout their life because they're not able to sort of pass salt, chloride, through their tissues, and therefore they build up mucus and they have lifelong infections and require therapy. It affects their pancreas and liver also. And what we've done is we've taken from an individual with cystic fibrosis, we've took their skin cells, we made these induced pluripotent stem cells, we've corrected the mutation, and now we've turned it into lung tissue in the laboratory. And the idea is to try to convert it into cells that we can actually deliver back to patients for curative purposes. So I've just given you an idea whether we start with sort of stem cells as they normally exist in the body, or cells that we basically induce and sort of create in the laboratory, all with the goal of regenerating healthy tissue for individuals who are diseased. And I think I'll stop there and let us go to questions.
Thank you so much. This is a lot of information to take in. So it's like a tabula rasa, where you're taking something from you and then erasing and then redesigning what can go back in. Yes. So thank you. So we have a plethora of questions. So I'm going to start with this. This question comes from a young woman. When I had a baby, I was given the option to store the cord blood in case my child ever needed the stem cells for a future therapy. Of course, we agreed to this, but what could this be used for down the line?
Very good. So I think at the present time, the main potential use of those cells would be in the case I was describing in terms of cancer. That is, if at the time that the child was delivered, that there was no cancer cells present in the umbilical cord blood, then that means at some point in the future, if they were to need a level of chemotherapy, that they would need a stem cell transplant afterwards, then at least the blood cells that are present would serve that purpose. And in fact, there's at MD Anderson and other places in the area, they actually use umbilical cord blood for that kind of transplantation. It's also potential that maybe they could be used for a sibling if they were closely matched, having those cells already available. There's also potential that there are other cell types present within the blood that may be useful as well, but I've never seen any I guess there are people who are deriving these mesenchymal stem cells for use in anti-inflammatory behavior, so potentially they could be used for that as well.
Thank you. We just got a question regarding retinitis pigmentosa, wondering if there are any treatments that you know of at this time for RP.
You know, I think actually this is one of the areas that has the greatest potential for within the next 3 to 5 years seeing some kind of potential. And the reason why I'm saying that is there are already trials going on. Oh, very good. So retinitis pigmentosa is a disease that affects particularly cells in the eye, but not the photoreceptors, I believe, and not the neural connections, but rather it's the epithelial sheet at the back of those cells. And patients who are born with inherited mutations in that area, that it's not able to provide the appropriate support to the other cells in the eye, and therefore people become blind or become severely disabled. In some sense, it's related to macular degeneration, which occurs, for instance, in many individuals as they age, where that same epithelial sheet basically deteriorates over time, and because it's not able to provide the support to the photoreceptors, that one begins to lose central vision. So, in fact, people are now using these induced pluripotent stem cells in the case of macular degeneration to turn them very efficiently into these retinal pigment these epithelial cells, retina epithelial cells, and actually doing trials already in Japan to deliver to try to restore sight. In the case of retinitis pigmentosa, either what you'd need to do is use cells derived from another individual where the cells are healthy, or people are doing genetic therapy. They're actually going in and using very precise genetic correction and then giving those cells back. So I think that's an area that offers promise.
And a similar question with a very different answer would be for macular degeneration.
Yes. So I think,
how are we doing?
So I think, what I'm most aware of is that there's at least one group in Kobe, Japan, who using these induced pluripotent stem cells has very efficiently turned them into these retinal epithelial cells and sort of delivering them as a sheet of cells right into the back of the eye. And it turns out you'd think, well, the eye is a very precarious place to introduce cells into, but in fact, that there are techniques available that are quite efficient in doing that. And what they've shown is that, at least first in mouse models, that they were able to correct vision. I guess there must be ways of telling whether a mouse can see or not, depending upon where the food's placed, or maybe their behavior in a maze, but essentially restored some activity there, and that's why they were able to move into the clinical trials with humans.
Thank you. Can you discuss what the half-life or the length of time that a stem cell therapy would be of benefit? Is there a law of diminishing return with a cell? Is there a natural cell death? So someone has a stem cell therapy, and I'm sure it depends on what the therapy is. Mm-hmm. So can we talk about how long it lasts? Or
Sure. In the case of the blood system, we know now that there are individuals out 3, 4, 5 decades after having received stem cell therapy through bone marrow transplant that are still have very, very functional blood systems with no evidence for decline. So it suggests, at least with that blood stem cell, when it's delivered into the right environment, that is back into the bone marrow, that it's able to regenerate cells, but it's also able to create more copies of itself, so that it's an ongoing process. For some of these other organ systems that we're talking about trying to treat with these stem cells, it may vary. I think it all depends. Stem cells don't exist in isolation. They are like a plant in a well-fertilized soil. Okay, they need the fertilized soil of the environment around them in order to be healthy, in order to divide, and to give rise to their progeny, or the cells that emerge from them. So I think a lot of these organ systems, when we think about delivering them cells in, we have to sort of find out ways to sort of incorporate those cells into the architecture. And I think there's a lot of work going where people are trying to do this now for things like Parkinson's and areas where we know what particular type of neuron needs to be replaced. But we don't really yet know effective ways of how to deliver those neural stem cells into the brain so they'll be taken up by the architecture and form the kind of networks and receive the kind of support to be healthy. So I think we're sort of an early stage, especially when we're talking about tissues with well-defined architecture.
And again, this sort of piggybacks on some of the other questions regarding stroke and other cerebral issues. So can you talk a bit about stem cell therapy in use of stroke? Or number one, is it happening now? And number two, if not, how close are we?
Well, it turns out that at UTHealth, we have a colleague, Dr. Sean Savitz, who has really been a pioneer in this area for a number of years, being a specialist in stroke, but then also delivering different types of cell from the bone marrow, and trying to enable the sort of the neural system, the brain, to be able to recover. Usually in stroke, there's going to be damage and there's going to be buildup of blood oftentimes, sort of a clot. So in order to be able to sort of regenerate those healthy neurons, one has to sort of contain the inflammatory processes, try to remove the clot. So, he's been doing sort of a progressive series of FDA-approved studies to basically look at individuals who receive the treatment and control, so that he can actually assess progress. And based upon those results, he's been allowed to move forward. I think that's one of the things that you'll find, I think, sort of being blessed by being here in proximity to the Texas Medical Center, is that, the strong academic medical centers like UTHealth and wherever, we focus on sort of doing scientific-based, careful, progressive, controlled studies to really figure out whether something is going to work, and if so, what are the mechanisms by which it's happening, and then take it to the next level. And I think that I can't overemphasize that. It means that in some cases the progress is slower than one would ideally like, but I think the important thing is that that's why our medical system and our biomedical research system is really the gem of the world, because there's that stepwise progression of very, very careful research translated into therapy. So it's a long-winded answer, but I'm trying to say that really, Sean, the work he's been doing, that he's been able to attract patients into that trial, delivering these cells either through the blood system or now into the brain.
So how does the average patient know what is an FDA approved? I mean, even our own primary care physicians who are in private practice may not be as up to date on what the cutting edge treatments are. How does someone with degenerative disease or arthritis even know if there is a therapy that is legitimate?
I mean, when I receive inquiries from diseases that I'm not familiar with, I usually start off by going, the NIH sponsors a website, I think clinicaltrials.gov, and that's usually a pretty good idea to find out at different centers around the country what is going on in specific areas. And then if I wished to know more, then I would usually list sort of the contact individuals there to sort of follow up and try to get as much information as I can. But I think that, in most cases, we're sort of at a relatively early stage in most of these. I think the case I talked about, blood stem cells for chemotherapy, that's been in existence, so there's quite a record there in terms of experience. I think some of these newer ones, we're sort of still on the initial stages. Great.
This next question is about plasma-rich platelets. How is there? It's a treatment that, from what I understand, many people are utilizing for joint issues. Does this have anything to do with stem cells? So when someone has PRP,
It's different.
Okay.
So it turns out that normally what you're doing is you're delivering to the individual sort of a fluid or a solution that's highly rich in platelets. Platelets are they're cells without nuclei. They're sort of that basically are involved in sort of wound repair. So they do have sort of beneficial properties in that way. But essentially, at least I would think that absent stem cells and absent sort of a cell delivery, it's going to be, you know, it'll be short-lived, I would think. And it may provide benefit because there may be certain nutrients that will sort of be helpful in that regard. But I think it's different from this kind of stem cell therapies we're talking about now.
Okay. Next question is, how close are we to growing whole organs that can be transplanted into a patient instead of having to wait for a donor? And it's okay to talk in decades, sir.
Yes, well, you know, growing an organ is quite a challenge. And the reason why I say that is if we, most organs actually have, you know, multiple cell types present within them. Just for example, most organs have some kind of blood flow that goes through them because it's through the blood that their nutrients are released then to the surrounding cells that keep the organ healthy. So there have been some initial studies done, particularly in mouse studies or rat, where people were able to take, let's say, a heart, and then treating it with sort of mild detergent and enzymes, they were able to essentially remove all of the cells that were present. And all you had was sort of this very willowy sort of with sort of white floccular material that had no cells in it. And what they then began to do is in a bioincubators begin to add cells back, and were able to sort of regrow the organ without all the cell types present, but still had the structure of the mouse heart, and then gave it back to a mouse, and the mouse, they connected it and was able to live a few hours. And that was the step forward. And they've done something similar for the lung. Dr. Just blanking on the name. She's at Texas Heart, and she leads their regenerative medicine. So she was known at University of Minnesota for essentially creating decellularized hearts and then regrowing them in this way. So I know there's active work going on, but it's quite a challenge to be able to translate that. Hopefully someday we'll be able to see it, because there is quite a demand for organ donors.
Great. Let's see. In preparation for this event, this is someone else. I read that in 2013 there were about 70 stem cell clinical trials under review. Yet here we are in almost 2019, and other than bone marrow transplants, all of these are still in the lab. What takes so long? And I believe the second part of this question had to do with, would it ever be advisable to leave the country for this kind of therapy?
Yes, let me first answer the first question if I can. I think, you know, as biomedical researchers, I think our first obligation is to do no harm. Okay, so I think that when we think about taking something that we've shown first in the laboratory, let's say in a tissue culture dish, and then we basically apply it in some kind of small animal model, the types that I've been talking about, maybe a larger animal model, then essentially then trying to translate it in a preclinical scale-up, quality-controlled way, where we then begin to do the carefully controlled trials, it is time-consuming. Each step takes both significant resources, takes new ideas, and then when one begins to first do the phase 1 safety trials and then move into phase 2, it is very involved. In some cases, it's more involved than, let's say, trying a small molecule drug that you may be trying to study for reducing blood pressure, and even in that case, that's a very long process. But I think where we're talking about delivering cells to try to repair, it is a long process. I think the second question regarding going abroad, I think I would have to say that this is something that is a concern to us in the community who work both as scientists and medical doctors. We understand, I think, that when someone has some kind of degenerative condition or something that's chronic and affecting, that there really is a desire to try to find something that'll help, just anything that'll help. And I think that and there's really been a proliferation of what we call sort of stem cell tourism, where people go to other countries to try to find help. And rarely are those cases any kind of sort of quality controlled or controlled studies that one knows that if one does have a benefit, whether it's a placebo effect or whether it's really due to the intervention that was done. There's also a question about safety. And just to give you an example, there was this is something that actually the FDA is very, very concerned about, not only what happens in foreign countries, but also what happens in our country, that there's sort of a proliferation of these kinds of clinics. And the FDA is not just reading I was just reading this morning, the head of the FDA, they're not really concerned about delivery into the joints, let's say, to try to restore knees, because that's been proven to be safe and there's really not been any kind. I think they're more concerned about things where it's going into the eye, going into the blood system, or going into the brain. So I think that probably depending upon what's being proposed, that just sort of raises the bar in terms of what one should be looking for in terms of qualifications of the people doing it, and what track record, what record is there that this is going to provide some kind of benefit.
We've had a couple of, I'm going to have just a couple of more questions, and then we will probably close off the evening. And of course, for our live audience, you are invited to stay. And for the web audience who's watching us, this will be up for about a week, so you'll be able to revisit. But we have had a couple of questions about the legality of stem cells. I think there's still such a confusion in the public about what is quote-unquote legal, and if you could address that. There's probably still some confusion about the use of embryonic stem cell lines versus our own adult stem cells. So, as I said, we've had 2 or 3 questions on that. So, if you could advise us legally.
Yes, let me see how I can sort of address this. I think that with respect to these pluripotent stem cells, I think that with the use of the induced pluripotent stem cells that I spoke about, there's no concern. Okay, that is, there's no legal issues surrounding that. When someone goes ahead and donates, for instance, just a small section of a skin biopsy or blood to make the iPS cells, one already gives authorization to turn them into these induced pluripotent stem cells, and they can then be used in that way. So I don't think there are any issues there. I think regarding work with human embryonic stem cells, I think different sort of states have come down differently in terms of how they're going to regulate that. The National Institutes of Health, what they've required is if you're going to receive research funding from them to work with human embryonic stem cells, that you have to, and use the funding for that work, you have to use one of their approved lines. And what they've done in terms of approved is they've made sure in that case to go back to, let's say, the woman who donated the original egg or the fertilized embryo. They've gone back to get all the appropriate authorization where that individual has given permission to turn those cells into embryonic stem cells and to be used that way. So I think I can't think of any really legal issues regarding the use of the adult stem cells, except this issue of regulation. I know Texas has taken some steps recently through legislation to sort of make it a little bit easier for companies to be involved in this, and I think this is something the FDA is taking a close look at to make sure that it's still, to what extent should the FDA be involved. Again, looking out for the health and safety primarily of the population. That's what the FDA takes very seriously.
Great. Thank you very much. This last question, let me add one of my own. We do have folks who have asked about this, people who would like to be part of clinical trials regarding stem cells. Would you also advise that they go to either their primary care provider or go to the NIH site on the clinicaltrials.gov to see what is available?
I mean, I think both would be very well worth doing. I know that there are ongoing clinical trials in the Texas Med Center on a number of fronts. I know that they're taking place around the country, and so I think that probably if I were faced with a disease that I wish to explore stem cell therapy, if I were not in the field, I think what I would probably do is, number one, reach out to my primary care physician who's most familiar with the status of my disease and whether he or she thinks that I might benefit from a stem cell therapy. But I think I also would go to, not all physicians who work in clinics are as familiar with what may be going on, so I think I would probably also go to the clinicaltrials.gov and look and see. They have a very good search engine where you type in what your disease is, and what you're interested in, and it'll pull up those that are relevant. Not all of them are stem cell related. Some of them are testing new drugs and the like. So I think there are resources available, and I think also, most individuals, if you do some searching in now it's so readily accessible through vehicles like PubMed or certain search engines where really the public can, the NIH requires that if you receive funding from them to do research, that anything you publish has to exist in a form on the web so that the public can have access to it, that it's not a restricted knowledge base. And you may need some help in terms of determining how to interpret what you're seeing, but I think the idea is that I think an educated patient and educated patient's family is really the best in looking for some type of therapy.
Okay. Thank you so much. This was so informative. And I believe at this point, I'm just going to thank you, Dr. Davis, for giving us much-needed overview of a complicated but truly exciting area of science and medicine, and for culling the fact from the fiction and the fiction from the fact regarding the present and the future of stem cell therapies. I would also like to thank Dr. Naoki Nakayama, who is sitting next to me, who has not had a chance to do anything but look down and type, for being so diligent in responding to the online questions that we just couldn't get to this evening. And certainly a huge thank you goes to our kind and generous hosts at Mercedes-Benz of Houston Greenway and ABC 13 for giving us a platform to supply our Houston community with the knowledge and awareness that we need regarding our health. I'd also like to extend our gratitude to UT physicians who are our partners in health, our clinical faculty from McGovern Medical School at UTHealth, who teach and train and treat and provide the highest quality of care for the entire family. In fact, the Dean of McGovern Medical School is with us this evening, so thank you very much. She's checking our homework, I have a feeling, but so tonight was the 6th of 7 UT Health House Calls. Our final one will cover healthy aging on Thursday, November 15th at Mercedes-Benz of Clear Lake. And of course, it will be available on the web. For more information about UT Health House Calls, please visit the Health Check section on abc13.com. And thank you all, and we hope to see you next time.